A very good afternoon, everyone. It is such a pleasure to be part of the FAScinate series and share with you insights into our golden crop, the oil palm, which is also a controversial crop under the spotlight of sustainability. Now, when you hear the word oil palm, people often think of it as the villain or even the Lord Voldemort of the plant kingdom. The oil palm has been accused of endangering our iconic species, and it cannot be denied that large-scale cultivation of oil palm has displaced the habitats of several threatened species. It has to be acknowledged that we have to stop clearing our precious rainforest. However, it is also important to get all the facts right and the full picture through an unbiased lens. The biodiversity hotspots where oil palm thrives coincides, if you look at this is where oil palm thrives. It coincides with regions of extreme poverty. Oil palm cultivation has been a major vehicle for rural socio-economic development. It has provided job opportunities and transformed the lives of millions of people. It was estimated that the oil palm industry had lifted 2.6 million rural Indonesians from poverty this century. While oil palm cultivation is often associated with large plantation companies, smallholders in fact account for 30 to 40% of global oil palm cultivation. The world population is projected to grow to 9.7 billion by 2050, and this will be paralleled with an increase in global demand for resources, including food and energy. Which crop do we turn to meet this increase in demand for oils and fats? Without doubt, it is the oil palm, because it is the most productive crop in the world. It is 6 to 10 times more productive than soybean, sunflower, and rapeseed. And if you think of it, it is 10 times more, and that means that soybean cultivation will require 10 times more land to produce the same amount of oil that the oil palm does. The oil palm is also very unique in that it is the only crop that produces two types of oil. The mesocarp, or the fleshy part of the fruit, produces what we know as palm oil. And the nut oil, or the kernel, produces palm kernel oil, which is very similar to coconut oil, and it is used mainly for non-food applications like soaps, detergents, and candles. Planted on just 7% of the total land area occupied by oil crops, oil palm contributes to 36.5% of global vegetable oil. All other crops would require substantially more land to meet the increased global demand. Soybean occupies 43% of the total land cultivated with vegetable oil, but produces only 27.4% of the global vegetable oil. Oil palm is thus the most sustainable choice because it has the best land use efficiency, but it has to be acknowledged that it needs to be produced more sustainably. There is a need for further increase in land use efficiency. Genomics, which is a highly precise biotechnology tool, can be leveraged for this purpose. Agricultural genomics, which is the application of genomics-based tools to improve crops and livestock, has ushered in the new green revolution. Genomics investigates and explores information contained in the genome, which is the genetic blueprint of life. The genome contains the entire set of genes responsible for every trait, from the color of your eyes to the texture of your hair, and even whether you can twirl your tongue. The genes are coded in DNA molecules and packaged in chromosomes. DNA molecules consist of 4 bases: adenine, cytosine, thymine, guanine. You can think of these bases as alphabets. The genome can have billions of these alphabets. The sequence and order of these alphabets makes up the genetic code. The human genome comprises 23 pairs of chromosomes. We were interested in breaking the genetic code of the oil palm. The oil palm has a complex genome of 16 pairs of chromosomes and about 1.8 billion base pairs, or the alphabets, which I just mentioned. Unraveling the genome was thus a challenge. However, with our collaborators, we successfully decoded the oil palm genome. It has more than 38,400 genes and is an invaluable resource for crop improvement and discovery of genes of important traits. Our decoding of the oil palm genome was published in Nature. We discovered 3 genes impacting yield, shell, fruit color, and clonal abnormality, and I will briefly describe each of these. First, the shell gene. We were especially interested in finding the shell gene because it is arguably the most important determinant of oil yield. The oil palm has 3 fruit forms: dura, which has a thick shell and therefore correspondingly less mesocarp and therefore less oil. The piscifera has got no shell and therefore should produce a lot more oil, but it is usually female infertile and does not produce fruit. It is used more as a male parent for pollen production. The tenera is a cross between the dura and the piscifera. It has a thin shell and hence more mesocarp and also more oil. Also, tenera palms tend to produce more fresh fruit bunches than the dura. Overall, tenera produces about 56% more oil than dura and 100% more oil than piscifera, since the piscifera does not bear fruit. Tenera is the commercial variety planted in Southeast Asia. However, there is often contamination with dura and to a lesser extent piscifera, leading to reduced oil and revenue loss in the tune of billions of ringgit every year. Fruit color is another important determinant of yield, as it is a measure of fruit ripeness. Palms are either nigrescence or virescence in terms of fruit color. Nigrescence fruits are either deep purple or black at the apex and undergo a minimum change in color upon ripening. The harvester on the ground finds it hard to tell when the bunch is actually ripe. Virescence, on the other hand, undergoes a dramatic change in color from green to orange when ripe. This makes it much easier for harvesters to determine when a bunch is ripe, leading to higher efficiency and yield. Now, besides the standard mode of cultivation from seed, tissue culture or cloning allows the mass propagation of thousands of genetic copies. This is a powerful shortcut to yield enhancement and could potentially increase oil palm yield by more than 30%. However, tissue culture often gives rise to abnormality. These are several of the abnormalities. These are several of the abnormalities. And an abnormality called mantled is the most common. Mantled results in abnormal fruit development, and the fruits often abort, resulting in major yield losses. It takes around 6 years by the time the palm or the clonal palm starts fruiting to know if a clone is normal or mantled. And by this time, it is really too late to uproot the abnormal palms. Because of the high risk, currently only about 1% of the total palms planted in Malaysia are clonal material, highlighting the need for a screening test before planting. However, in order to come up with a screening test, we first had to discover the cause of abnormality. The phenomenon of clonal abnormality was first observed in 1986, and for almost 4 decades, scientists in the oil palm industry was frantically looking for the cause. We knew from several of our studies that clonal abnormality is not a genetic phenomenon, but rather an alteration in the epigenome. Now, what is the epigenome? The epigenome is a second code written on top of the first code. So this is the first code, and this is the epigenome, or the second code. While it does not change the first or genetic code, as you can see there is no change in the genetic code, it acts as a power switch that can turn on or off the expression of the genetic code. It does this via various chemical tags, the most common of which are methylation, histone modification, and small RNA regulation. In methylation, methyl groups are overlaid on this is a methyl group, and it's overlaid on the genetic code. The environment, including nutritional status, plays a significant role in regulating the epigenome. For example, in honeybees, the development of a larva into a queen bee or worker depends on its nutritional status. Feeding the larva with royal jelly triggers epigenetic modification, including DNA methylation, which gives rise to the queen bee. Larvae destined to be worker bees are fed pollen and nectar. We knew from our early studies that the mantled abnormality involved changes in methylation, but did not know which gene was involved. However, once we had sequenced the all palm genome, we had this amazing resource, and we could study the methylation across the entire genome. Using 180 clonal palms representing the diversity of clonal all palm across Malaysia, we were able to pinpoint the exact DNA methylation change out of 1.8 billion bases. If you look at this is the genome, and different clones have got different methylation changes, but there's one change that was common to every single clonally abnormal palm, and we found that we pinpointed that causative DNA methylation change, right? And we found that the methylation status of a retrotransposon called karma. In layman's language, a retrotransposon is a jumping gene. A jumping gene called karma has inserted itself in the flowering gene, and the methylation status of karma is the one that causes mantled. If I show this a bit clearer. Now, if karma is poorly mantled, as in this case, poorly methylated, in this case, then you get the mantled fruit. However, if it is richly methylated, then you get a normal fruit. Okay? Our discovery of the role of karma in all palm clonal abnormality got into the front cover of Nature. And it says here, weed out bad karma, the epigenetic trait that can ruin an all palm crop. Our sequencing of the all palm genome and discovery of the shell gene also got into Nature, in fact, as back-to-back articles. And discovery of the fruit color gene got into Nature Communications. This was a first for Malaysian science because both the first and corresponding authors, as well as the majority of authors, were Malaysians for all these Nature publications. Besides the scientific recognition in high-impact journals, we also received recognition from the international press, including the BBC, New York Times, and The Guardian, which are usually highly critical of the all palm. But in this instance, they were highly positive. In fact, Scientific American interviewed me and Harrison Ford, who, besides being a very well-known actor, is also an activist who is highly critical of the all palm industry. However, he praised MPOB for its genome work and said that discoveries like Ver and shell genes could help the palm industry strike a better balance between growers and consumers because they address the elementary roadblock to proper palm industry management. Very importantly, our discoveries were translated into commercial outcomes, representing the world's first high-throughput genome-based diagnostics for all palm. The technology, which we trademarked SureSavit, was licensed by MPOB to Orion Bioscience, a Malaysian-based company. SureSavit Shell, SureSavit Ver, and SureSavit Karma allow screening for shell, fruit color, and clonal abnormality, respectively. One has to just take a leaf punch or small amount of DNA, small amount of material from the seed, extract the DNA, and analyze for the traits of interest. The technologies thus allow pre-selection of palms as early as the nursery or seed stage and allows for planting only desired palms, thus increasing the sustainability in existing areas. SureSavit technology was featured in BBC's Digital Disruptor series and chosen as one of the top disruptive technologies in the world. Using the SureSavit shell technology, MPOB tested 1.1 million seedlings, representing 93.5% of immature oil palm planted in Malaysia. We found that 12.8% of the palms were contaminants. This is more than 2 times the maximum contaminant level of 5% allowed by cerium. This is where the cerium standard is 5%, but the contamination rate, average national, is way higher than that. Now, of the 12.8% contamination, about 7.8% came from seed producers and 5% further down the supply chain. While the contamination from seed producers was probably unintentional, resulting from occasional self-fertilization or cross-fertilization from neighboring plots, the 5% contamination further down the supply chain came from fraudulent practices where they were added to the supply chain. Based on the potential gain in oil yield from shell testing, we estimate that Malaysia would see a 6.9% gain in land use efficiency and over $2 billion gain in annual income, emphasizing the importance of shell testing to Malaysia's economy and the sustainability of the oil palm sector. It highlights the power of technology. However, one of the biggest challenges is overcoming barriers to adoption of technologies and the Malaysian mindset of short-term profits over long-term benefits. We must be ready to embrace change and pay for technology. With that, I'd like to acknowledge a fantastic team that I've worked with. I was really fortunate to work with an amazing team, if I could have them here. These members are whom I consider friends and family. I would like to especially highlight Dr. Rajinder Singh, who is the head of the genomics unit, Dr. Melina, who was the head of the breeding and tissue culture unit at that time, Dr. Leslie Lau, the head of the bioinformatics unit. Now, one of the major ingredients of MPOB's genome success story is that we have the world's largest oil palm germplasm collection. Credit for this goes largely to Dr. Rajanaidu, who made several expeditions to various parts of Africa, South and Central America to collect these wild accessions of oil palm. We have always believed that science cannot thrive in isolation and fostered collaborations with world-class centers. Our main collaborator for the oil palm genome program is Orion Genomics. This is a great team. I would like to especially acknowledge Nathan Lakey, the CEO and president of Orion Genomics, and Rob Martinson, the professor of plant genetics at Cold Spring Harbor and co-founder of Orion Genomics. The MPOB genome program would not have reaped the success it did if not for its visionary leadership. I would especially like to acknowledge Dr. Dato Ahmad Parviz, the current DG of MPOB, who has been a great champion and supporter of this program. The oil palm genome program was, in fact, first started or initiated during Dato Muhammad Basri's tenure as DG. And a special thanks to Dato Chu Yun Mei, The oil palm genome sequence was published and shared with the international scientific community during her tenure as the DG. I would like to re-emphasize that the work I described was a team effort led by MPOB. To all my colleagues and collaborators whose individual brilliance has contributed to the success story I just described, thank you. I would like to end with a quote from Franklin Roosevelt. I'm not the smartest fellow in the world, but I sure can pick smart colleagues. Thank you.